Hello students, how are you? Are you fine? Had your dinner? So in the last class we have discussed about classification of kingdom Monera, where we have discussed about Archaebacteria and Eubacteria. So today we are going to start discussing in detail about Eubacteria. So what are Eubacteria, students? Eubacteria are nothing but true bacteria. True bacteria. Why they are called true bacteria? Because they have cell wall material which is called murine. So what is the name of cell wall? Murine. Cell wall material is present, and murine is also called peptidoglycan. Peptidoglycan. This is the another name of murine cell wall. Since they possess the cell wall murine, they are called as true bacteria, and most of them are not capable to cope up with extreme conditions. Okay, so they are not extremophiles. So where does this Eubacteria live? The Eubacteria live in soil, in water, and also inside our body and outside our body, and lot of them also cause diseases in human beings and plants. So now let us discuss about different types of Eubacteria. First is cyanobacteria. What bacteria? Cyanobacteria. Cyanobacteria is also called blue-green algae. These bacteria are also called blue-green algae. So why they are called blue-green algae? They occur in the colors like blue and greenish color. That's why they are called blue-green algae. Actually, they are not algae, but it is a nickname given to this bacteria as blue-green algae. So where this cyanobacteria live? Cyanobacteria, they are found living in fresh water such as ponds and lakes, and also found in marine water, that is in the ocean, marine water, and also found living in moist soil. Okay, so in these three places we can see these bacteria living. They are terrestrial as well as aquatic. They can live both in the land as well as in the water. Okay, and what is the shape of this cyanobacteria? Some are unicellular. Some are colonial. Some forms colonies. Colonial. So what is colonial? Several single-celled cyanobacteria will form a group and live as a colony, as a group. So that type is called as colonial. And some are filamentous. What is filamentous? Some of the cyanobacteria, they form chain-like structures and they resemble long threads, which are called as filamentous. Okay, so we can find unicellular, colonial, and filamentous form, three forms. So they have an important pigment which is responsible for photosynthesis. Chlorophyll A pigment is present in this cyanobacteria. This is one important character of this bacteria. So because of this pigment, these bacteria are capable to carry out photosynthesis. So by using this chlorophyll pigment A, cyanobacteria carry out photosynthesis. It is called oxygenic photosynthesis. So what is oxygenic photosynthesis? It is a photosynthesis process which involves release of oxygen. It involves release of oxygen into the water. Okay, so once oxygen is released into the water, what happens? The water gets oxygenated. All other fishes, they can happily use the oxygen for their respiration, and some of the oxygen obviously will escape into the atmosphere. Okay, so this is a great benefit which the cyanobacteria does to the environment, that is releasing oxygen into the environment. Okay. Next, what are the examples of cyanobacteria? Students, look here. Nostoc. Next is Anabinae. These two are photosynthetic cyanobacteria. They can carry out photosynthesis. And one more example I am giving you, that is spirulina. So these are the three examples important for your NEET exam. So you can see the cyanobacteria nostoc. This is the photograph of nostoc cyanobacteria. Look here. It is looking like a long filament made up of several single cells attached to end by end to form a long chain like structure. So this is what is called as nostoc. Okay. Here you can also see there is a specialized cell which is comparatively larger in size. Okay, I will tell about it. So now we are going to discuss the structure of cyanobacteria called nostoc. Now I am going to draw the diagram of nostoc. Students, you copy down in your notebook nostoc. So what is this nostoc? Nostoc is a filamentous. It is a filamentous photosynthetic nitrogen fixing cyanobacteria. So it is having a filamentous shape and it is capable to carry out photosynthesis, and also it can carry out nitrogen fixation. That means convert the atmospheric nitrogen into ammonia. We call it as nitrogen fixation. Now let us see the structure. This is one cell of nostoc. This is another cell. Several cells are attached end to end to form a chain. So this is what is called as nostoc cyanobacteria. Okay. So all these are individual cells attached end to end. So here you can see a specialized cell comparatively larger in size. So this cell is called as heterocyst. It is called heterocyst. What is the function of this heterocyst? It is involved in nitrogen fixation. It carries out nitrogen fixation and also stores the fixed nitrogen in the form of ammonia. Okay. The entire filament of nostoc is covered by a slimy material which protects the bacteria. This slimy material is called gelatinous sheath. What is called, my dear students? It is called gelatinous sheath. Gelatinous sheath. The gelatinous sheath is made up of a material called mucilage. A slimy material called as mucilage. And what is mucilage? Mucilage is chemically, it is a polysaccharide. Chemically it is a polysaccharide. Okay. So this entire filament will be green in color. All these cells will carry out photosynthesis except this heterocyst which will carry out nitrogen fixation. The remaining cells are called as vegetative cells. They are called vegetative cells. The function of vegetative cells is photosynthesis. Note down, students. Draw this diagram in your notebook. Come on. Completed? Shall I rub the board? So now we will discuss some more points about this cyanobacteria. So just now we have discussed about the cyanobacteria called nostoc, another cyanobacteria anabinae. These two are beneficial to farmers. Both of them are used as biofertilizers. They are used as biofertilizers in paddy fields. So farmers, what they do, they will apply this cyanobacteria in their fields, in the paddy fields. So they will carry out nitrogen fixation and increase the fertility of soil. Okay. So next, what is another example which we discussed? Spirulina. Spirulina is used as single cell protein. SCP stands for single cell protein. Spirulina is rich in proteins and it is used as a protein supplement, and nowadays it is used as a facial also in the beauty parlors. Okay, because it is packed with a lot of nutrients like proteins and vitamins, all the stuff. There is a cyanobacteria which grows in the ocean that is called Trichodesmium. Trichodesmium erythraeum. This is a scientific name of a particular variety of cyanobacteria. This is living in marine water. This is found living in the marine water, and this is known for causing Red Sea. It causes Red Sea. It is known for causing Red Sea. That means the entire ocean appears red in color because of excessive growth of this cyanobacteria called Trichodesmium erythraeum. Okay, so this cyanobacteria, when they grow excessively in the water bodies, the entire water surface will be completely appearing in green color. So we call it as algal blooms. Simply we call them as blooms. Okay, you can see the video of algal blooms here, students. So this is the blooms of cyanobacteria which are formed on the surface of water due to excessive growth of this bacteria. Why this bacteria grow excessively? When the water is having lot of nutrients, so that will favor the bacteria to grow excessively like this. Okay, copy down this. Students, I am going to rub the board. So now put heading chemosynthetic autotrophic bacteria. Chemosynthetic autotrophic bacteria. What are these chemosynthetic autotrophic bacteria? These bacteria, they obtain energy. From inorganic compounds like NO2, NO3, ammonia, etc. Okay, so they use the inorganic compounds like nitrate, ammonia in order to obtain the energy. So these bacteria, they oxidize inorganic compounds and use energy to produce ATP. What is ATP? ATP stands for adenosine triphosphate. It is energy currency which is produced in the cell. So these bacteria, what they do, they will use the inorganic compounds like this. They oxidize these inorganic compounds and use the energy which is obtained during oxidation to produce a molecule called ATP. Okay, so examples of chemosynthetic autotrophic bacteria: Nitrobacter. Nitrobacter, example of chemosynthetic autotrophic bacteria. These bacteria also involved in recycling of iron, phosphorus, next nitrogen and sulfur. So these bacteria also, they are involved in recycling of elements, mineral elements like iron, phosphorus, nitrogen and sulfur. Okay, this is about chemosynthetic autotrophic bacteria. These bacteria can prepare their own food material, but energy is not from sunlight, the energy is from the inorganic compounds. Okay, copy down students. हो गया, मिटा दूं, चलो। Now, heterotrophic bacteria. Heterotrophic bacteria. Heterotrophic eubacteria are those bacteria, they obtain their food material from other sources. They cannot prepare their own food. So, what are heterotrophic bacteria? They obtain nutrients. They obtain nutrients from other sources, cannot prepare own food, cannot prepare own food. Such bacteria are called as heterotrophic bacteria. So now these heterotrophic bacteria are most dominant bacteria. They are the most dominant bacteria. So more type of heterotrophic bacteria are found in the nature. They are the most dominant bacteria in nature. So this heterotrophic bacteria includes parasitic bacteria. Parasitic bacteria, they are those heterotrophic bacteria that live inside the body of other organisms. They can live inside the human beings, inside the animals as well as plants, and they cause number of diseases. So parasitic bacteria, they live inside other organisms, and cause diseases. So examples of parasitic bacteria we'll discuss. The first one is Salmonella typhi. This particular bacterium causes typhoid fever. It causes typhoid disease. Okay, this is the name of the disease. Next, Clostridium tetani. This particular bacteria causes tetanus disease. Okay, next, Vibrio cholera causes cholera disease. So these are all the diseases which are seen in human beings. Now we will discuss about a bacteria which cause disease in the plants. Xanthomonas Citri. This causes a disease called citrus canker. Citrus canker. So come on students, note down this. Students, heterotrophic bacteria not only includes this parasitic bacteria, some are useful bacteria. Now let us discuss some useful bacteria. Useful bacteria. Useful bacteria. So examples of useful bacteria. Lactobacillus. What is that? Lactobacillus. This is used for making curd. This bacteria is used for making curd from milk. So this is one of the useful bacteria. Okay. So next, Rhizobium. Rhizobium. This bacteria is useful for nitrogen fixation. It is a symbiotic bacteria. Symbiotic bacteria in legume plants like groundnut, which help in nitrogen fixation. So like this, the Eubacteria also includes useful bacteria like Lactobacillus and Rhizobium. And there is another bacteria which is called Streptococcus. Streptomyces. This particular group of bacteria produce variety of antibiotics which are used to treat bacterial infections. Streptomyces group of bacteria produce antibiotics which are used to treat bacterial infections. Okay, understood? Copy down. So I'm erasing the board. Now we will discuss about mycoplasma. Mycoplasma bacteria are also called PPLO. So what is PPLO? PPLO stands for Pleuropneumonia like organisms. Pleuropneumonia like organisms. Mycoplasma. So this mycoplasmas are PPLO bacteria. They are smallest bacteria in the kingdom Monera. They are the smallest bacteria. And what about their size? Their size ranges from 0.1 to 0.5 micrometers. 0.1 to 0.5 micrometers. Very small, smaller than normal bacteria. Okay. And one more character here. Cell wall is absent. What is absent? Cell wall is absent. So because of absence of cell wall, these bacteria have irregular shape. The bacteria have irregular shape. And often they change the shape of their body. That's why these bacteria are called pleomorphic. Pleomorphic, which means the bacteria which often change their shape. Okay. Next, these bacteria can survive in absence of oxygen. They can survive in absence of oxygen. So even without oxygen also these bacteria can survive, and they are parasites in plants and animals. They are parasites. They cause diseases in plants and animals. Plants as well as animals. Since cell wall is absent in these organisms, they are not only pleomorphic, they are resistant to penicillin antibiotic. So these bacteria cannot be killed by penicillin antibiotic. They are resistant to penicillin antibiotic. Why? Because penicillin antibiotic will act on the cell wall material. It inhibits the cell wall formation in bacteria. That is the reason why the penicillin antibiotic cannot kill your Mycoplasma bacteria. Now let us see what are the diseases caused by this Mycoplasma. So Mycoplasma will cause sexually transmitted disease. Sexually transmitted disease in man, it is called urethritis. In women it causes reproductive tract infection. So what is the Mycoplasma bacteria which causes this sexually transmitted disease? The name of that bacteria is Mycoplasma genitalium. So this is the Mycoplasma bacteria which cause the sexually transmitted diseases in men and women. Okay, and in plants it causes little leaf disease of brinjal. So what are the points of Mycoplasma, students? They are the smallest bacteria ranging from 0.1 to 0.5 micrometers. They are also called PPLOs, and cell wall is absent. They can survive in absence of oxygen, and they are parasites in both plants and animals. Okay, so and they are also resistant to penicillin antibiotic. Come on, note down this, students. Note down. हो गया? चलो. Next, actinomycetes. Actinomycetes bacteria are very different. They are called by name called ray fungi. This is a nickname given to this bacteria, ray fungi. Why they are called ray fungi? Because they have their body which is similar to fungi. So just like the body of fungi, they have a filamentous structure. That's why the name ray fungi. And also they behave as fungi. They are saprophytes. They are saprophytic bacteria which grow in the forest floor. Where do they grow? They grow in forest floor. So what does they do in the forest floor? They will decompose the plant waste in the forest floor. So imagine this is the forest. Lot of trees are there in the forest, right? In the forest floor, what you can find? In the forest floor, you can see lot of plant waste like leaves, fruits, branches, flowers. These are all decomposing in the forest floor. So these people, actinomycetes bacteria, they grow in this forest floor, decomposing the plant waste. So they help in humus formation. Ecologically, they help in humus formation, and humus is good fertilizer for the plants. Humus is going to be a good organic fertilizer for the plants. So they help in humus formation. So actinomycetes bacteria have cell wall called as mycolic acid. What is the cell wall material? Mycolic acid is the cell wall material present in actinomycetes bacteria. Now examples of actinomycetes is Streptococcus Streptomyces frankia. So these are the examples of actinomycetes. Copy down, students. Put heading reproduction. Reproduction in bacteria. So students, as we discussed in the chapter living world, reproduction is seen in all the organisms except few organisms like mules and the female worker bees. Okay, so reproduction process is also observed in bacteria. Let us see how the reproduction will take place in bacteria. The first method is binary fission. Binary fission. It is a method of asexual reproduction. Look here carefully. What is a binary fission? So imagine that this is a bacteria. This is a bacteria. So inside the bacteria there is cytoplasm and there is nucleoid. This is a nucleoid which contains the DNA. Deoxyribonucleic acid. It is circular in nature. Okay. So first the bacteria is going to replicate this DNA. So one copy of DNA which is present inside the bacteria, it is going to double. Okay. So after some time, what happens in the same bacteria, 2 copies of DNA will be available. Okay. So replication of DNA takes place. The DNA doubles, 2 copies of DNA will be produced. Afterwards, the bacteria slowly starts dividing into 2 equal halves. The bacteria starts dividing itself into 2 equal halves. So this portion it separates as one separate bacterial cell, and this portion it separates out as another bacterial cell, and each newly formed bacteria will receive 1 copy of DNA. So these newly formed bacteria are called daughter bacteria. They are called daughter bacteria, not son bacteria, only daughter bacteria. Understood? This is called binary fission. It is a method of asexual reproduction. It is an asexual form of reproduction. Okay. So next, second method, spore formation. Spore formation. What is this spore formation? This process of reproduction is especially seen during unfavorable conditions. Unfavorable conditions such as high temperature or absence of food material in the surroundings. In such circumstances, the bacteria will use this spore formation method. So here what happens, the bacteria it produces a tough cell-walled structure called as endospore. What is this called? Endospore. Internally, the bacteria will produce a tough cell-walled structure called endospore. Inside this endospore there will be cytoplasm and some amount of DNA. So this endospore is resistant to unfavorable conditions, to high temperatures. When favorable conditions return, this endospore, from this endospore a small bacteria will come out and it starts its life. Okay. So next, third method of reproduction, that is called conjugation. Conjugation. Conjugation is a kind of a sort of sexual reproduction. It is not a true sexual reproduction. It is a sort of sexual reproduction, means a kind of sexual reproduction, not a true type of sexual reproduction. So during conjugation, what happens? 2 compatible strains of bacteria will come close to each other. So one is called plus strain and another is called minus strain. Okay. So these 2 bacteria, now they are going to share the genetic material. The plus strain is considered as the donor. Okay. It will produce a tube-like structure called the pilus. By using this tube-like structure, it will transfer its DNA into the opposite bacteria, which is called as the recipient bacteria. It will transfer its DNA into the another bacteria. So it is considered as a primitive method of sexual reproduction. Bacteria will choose this method especially for the bacterial evolution process. Okay, so these are the three methods of reproduction. Students, understood? Come on, copy them in your notebook. Students, I have brought something for you. Can you guess what is this? What is this? This particular jar is full of bacteria. Guess what type of bacteria is this? Exactly, this is cyanobacteria spirulina. Okay, you can see here. You can see small filament-like structures inside. This is full of spirulina bacteria, autotrophic photosynthetic bacteria. Okay, so why it is green in color? Because this bacteria have chlorophyll A pigment, by using which this bacteria can carry out photosynthesis. You know, students, nowadays this bacteria is used as a protein supplement. So I just ordered online and brought this, and I am growing in my home spirulina. If you want this spirulina, ask me. Comment in the comment section. I will send you this spirulina cyanobacteria. Okay, so we'll meet in the next class, students. Bye bye.